Great, thank you. And thanks everyone for attending this IDG symposium. Today's focus is going to be on ion channels. And we have two great speakers today, Henry Colecraft and Markus Delling. I believe, Henry, you're on the docket first, so I'll give a brief introduction to you. Henry is originally from Ghana, West Africa, and I understand his father did his graduate work in Boston, so he took his family along. Maybe that was his first experience in the United States. And when he was a teenager, he moved to England, where he did his bachelor's degree at the University of London and King's College. He then moved back to the States, where he completed his Ph.D. in pharmacology at the University of Rochester in New York, and then went on to do his postdoc training in Johns Hopkins. And he remained there as an assistant professor in biomedical engineering until he was recruited to Columbia as an associate professor around 2007. He's an international leader in molecular physiology of ion channels, particularly those signaling pathways in the nerves and the heart. And his group has done some really, has contributed seminal advances to understanding molecular mechanisms underlying the regulation of voltage-dependent calcium and potassium channels and accessory subunits like the post-translational modifications, ubiquitination, signaling molecules. He's really unafraid to get involved with new technology and new approaches. And today he's going to give a little presentation on some of his work in nanobodies, I believe. Henry, it's a pleasure to have you. Thank you for joining.
So thanks, Mike, for that introduction. It's a real pleasure to be here. And typically, when I'm going to give this lecture, I usually say that the work I'm going to present is really useful for every type of protein, not just ion channels. But I don't think I have to say that with this audience, because we're all ion channel aficionados. But I appreciate the opportunity to share some of our work with you. We've been interested in developing nanobodies to engineer or to regulate ion channels for various activities. And I just want to share some of that work today with you. So the starting point of this particular story starts from the realization that there are several very serious diseases, starting cardiac death, cystic fibrosis, epilepsy, and the list continues, which on the very surface of it are actually quite different diseases. But further reflection, we realize that many instances of these diseases really have a common molecular mechanism in that the mutations or conditions lead to decreases in the functional expression of different ion channels that underlies these very different diseases. And so the simple big idea we had was if we could figure out a way to reverse this trend and made it general, then we could have a transformational approach that could be utilized for many different diseases. And so that's what we've done. And in order to introduce how we did that, let's look at the life cycle of a traditional ion channel or of a typical ion channel that we study. So these ion channels, shown here as ovals or ellipses, are born in the ER. They are trafficked to the Golgi where they are processed, and then finally to the plasma membrane where they are functional. From the plasma membrane, they can be removed and either recycled back to the membrane or taken to the lysosome where they're degraded. So what we realized early on was that this post-translational protein tag, ubiquitin, is actually quite important for various steps of the trafficking as well as the stability of the protein. So I can simplify this rather complicated diagram to show this, that the functional density of a typical ion channel that we study is driven by three main processes: the rate of delivery, the rate of removal, and the rate of degradation. And ubiquitination really biases the equilibrium towards retention and degradation. So the light bulb moment for us came from the realization that many mutant ion channels really behave like wild type channels with a lot of ubiquitin on them, in that they are retained and degraded. So the hypothesis, the very simple hypothesis we had going was that de-ubiquitination of such mutant channels could help restore their surface density, and if they were functional, lead to rescue of a disease phenotype. Of course, the problem is that ubiquitin is a global signal, is utilized to control the homeostasis of all proteins inside the cell. And so the problem really became, how do you specifically remove ubiquitin from a target of choice without disturbing global protein homeostasis? And that's what we've done. And we did that with this molecule, a bivalent molecule that we refer to as an engineered de-ubiquitinase, an NDUB. And what it is, it has a targeting moiety, which is an intracellular actin antibody, a nanobody, fused by a linker to a catalytic unit, which is really the cell's own natural erasers, the catalytic unit of a de-ubiquitinase or a DUB. So this design principle endows these NDUBs, for short, with certain unique qualities. First, we expect they'll be very highly specific due to the targeting moiety, and thus have minimal off-target effects. They are modular, so we can really simply switch out the targeting moiety and target different proteins and therefore different diseases. And we expect it to be highly potent due to the catalytic activity of the enzyme. So this is it in concept. How does it work in principle? Can we actually get this to work? So Scott Kanner is the graduate student who developed this in the lab and came up with a biochemical assay to test whether the NDUBs would act in the way that we predict. All right, so we're going to use a model ion channel, KCNQ1, which forms a tetramer, and we fuse to it YFP so that we have a handle that we can target with different molecules as well as have something to probe. What was known in the literature is if you co-express KCNQ1 with this E3 ligase NET4, it ubiquitinates it and causes its endocytosis and degradation. And so our idea was that this would give us a system to test whether we could use an NDUB, in this case using a YFP targeted nanobody fused to a catalytic domain of the de-ubiquitinase called OTAT1. Okay, so here's the data. Untransfected cells have no KCNQ1. This is a pull down of Q1 followed by a Western blot. And under control conditions where we just express a naked nanobody by itself, you get 4 bands representing the monomer, the dimer, the trimer, and the tetramer. If we co-express NET4L, the density of that band goes down, consistent with that protein being degraded. And we can strip this blot and probe with the ubiquitin. And what you can see is that there is some basal ubiquitination of the channel under control conditions, but with NET4L, you get a lot more, even though the amount of channel is down. So now what happens when you add the NDUB? We're gratified to see that we restored the protein expression, and that was due to really stripping ubiquitin of that protein. All right, so in principle, it seems to work, at least from this biochemical assay. The next thing we needed to test was whether the de-ubiquitinated channel made it to the cell surface and was functional. In order to test that, we came up with an optical approach that allowed us to simultaneously measure the total protein expression by looking at YFP fluorescence. And we had a surface epitope tag that we could probe with a fluorescent toxin to look at the surface population of channels with a red fluorophore. And then what we do is apply flow cytometry so we can measure quickly 100,000 cells or so, quantifying the red fluorescence, which is the surface, as a function of the total YFP, the total protein expression. And what you can see under control conditions is that YFP positive cells, there's much trafficking of the channel to the cell surface. When we co-express NET4L, we markedly diminish the surface density of the channels as well as degrade it, as indicated by a decrease in YFP fluorescence. When we add the NDUB, we completely rescue both the surface trafficking as well as the total density of the channel. Do they function? Well, we apply patch clamp to test that. So in the whole cell patch clamp mode using recombinant channels, we can measure the current that comes through all the channels from that single cell. Under control conditions, we record robust currents corresponding to Kc and Q1 at the cell surface. If you co-express net4L, that current is totally wiped out due to the endocytosis and the degradation of the channel. But with the NDAB, we completely recovered that back to wild type levels. Okay, so there were two important controls that we were concerned about before we tested the efficacy of NDABs in a disease setting. And those controls are shown here. First, this is what I've shown you before under the control condition, just represented in a different way, a cumulative distribution. So the gray is the red fluorescence intensity of the surface channel. And when we add net4L in the red line, it goes to the left because the channel is removed from the cell surface. In the presence of net4L, if we add the NDAB, we slam it back to the right because we recover the channel to the cell surface. So the first control was whether we required the DUB activity, the enzymatic activity of the DUB for this effect. And so we made a catalytically dead mutant. And when you do that, the blue curve no longer shifts. And so you require the catalytic activity of the DUB. The third was whether we actually required the targeting of the DUB to the channel of interest, because overall you're overexpressing an enzyme. So we could test that simply by leaving out the YFP. And then if you co-express the NDAB with it, you see that you no longer get the blue shift as well. And so this is totally critically dependent on the targeting to the target of interest. And so you're not getting non-specific effects. All right, so with that under the bag, then we turn our attention to a disease phenotype. And long QT syndrome is a rare disease occurring in 1 in 2,000 live births. And accounts for about 400,000 cases of sudden cardiac death, a significant portion of that in each year in the US. So 35% of long QT cases are due to loss of function mutations in Kc and Q1. And what this typically does is reduces the repolarization capacity of the cardiac action potential. That's causing a prolongation of it, which results in a lengthening of the QT interval in the electrocardiogram. And what's dangerous about this is that it predisposes these people to lethal ventricular arrhythmias to start your point, and then sudden cardiac death. So it turns out that many of those mutations cause a deficiency in the trafficking of the channel to the cell surface. And so we asked whether we could potentially use NDABs to rescue that by de-ubiquitinating the mutant Kc and Q1 channels. So what we did then was to essentially make a number of disease mutations, human disease mutations in Kc and Q1. And we used the surface staining assay that I've shown you before to test whether they inhibited the surface density of the channel. So compared to the wild type, which is this dotted line right here, you can see that many of those mutations, the red bars under control conditions, just expressed with the naked nanobody, had a substantial deficit in the trafficking to the channel surface. You can see that pictorially right here in that the wild type channels have a red staining on the cell surface. And this is totally gone in one of these mutations, G589D. What we're really gratified by was that when we co-expressed the NDAB, we actually rescued many of these to essentially wild type levels, shown right here for G589D. In addition to rescuing trafficking, they also appear to rescue the current. So here's one example for R591H, which completely almost eliminates the current compared to wild type. If you add a small molecule agonist for Kc and Q1, you're actually able to recover a little bit of it, indicating that some channels do get to the surface and can be regulated once there. And that's what we got with the NDAB for R591H. You get about 50% of the current just rescued with just that NDUB by itself. And this was interesting because for 591, you basically recover the whole current. And so what this indicated was that the mutation, in addition to inhibiting trafficking, also affected the gating of the channel. But now we could titrate that with the small molecule agonist and really get even a super rescue of the current. So all these data are done in hex cells, and it was important for us to test whether we could actually see this in actual heart cells. And so using guinea pig ventricular myocytes, which have endogenous KC and Q1, that gives rise to the slowly activating outward current known as IKS. If you co-express wild type Q1 in there, you get a nice IKS signal. And this is the normal action potential that you typically get. If we add in, by using a virus, a G589D mutant, we recapitulate the lengthening of the action potential and the decrease in IKS that is seen in human patients. And it was really great to see that when we express the NDUB with this, we recovered the outward current and normalized the action potential duration. So it seems to work, at least in this KCNQ1 disease phenotype. One of the things we were most excited about by this technology was the potential that we could, just by switching the targeting moiety, target this technology to different diseases. And so we tested the strength of that idea by looking at a different ion channel mutation, caused by a mutation in the chloride ion channels, cystic fibrosis. So this is the most prevalent genetic disorder in Caucasians, affecting about 85,000 people worldwide. And the symptoms of the disease are impaired mucus clearance from airway epithelial cells in the lungs. That leads to uncontrolled inflammation and recurrent infections that ultimately destroy the lungs and decrease life expectancy. The standard of care for a long time was antibiotics to treat infections and airway clearance approaches. And that led to the lifespan plateauing around 40 years, 45 years or so, 40 years or so. The disease is caused by mutations in the chloride ion channel, Cftr, which prevent it from getting to the cell surface. So very similar to what we saw with KCNQ1. Very recently, Vertex Pharmaceutical, using high throughput approaches, identified small molecules that could actually help to correct and traffic some of that channel to the cell surface, called correctors. And once there, they have potentiators that can have the channel open further. However, even with these new drugs, there's incomplete efficacy for many of the mutations. There's an unknown mechanism of action, so it's hard to improve upon them. And for many mutations, they're actually totally inefficacious. And so we wondered whether we could improve upon this with NDUBs. And so similar to what we did with KCNQ1, we made a channel with a YFP tag and a surface epitope tag that we could probe with the bungratoxin. We made an NDUB, and then we made several mutations representing 6 different classes of cystic fibrosis mutations that affected the folding trafficking or the plasma membrane stability of the protein. And you can see that compared to the wild type dotted line here, all of the mutations cause a substantial decrease in the surface trafficking of the channel. The red bars is what you get when you put in one of the FDA-approved small molecule correctors, Cftr. And you can see that it does actually help rescue some of these mutations better than others. In blue is what you get with the NDUB by itself. And it was great to see that the NDUBs were either similar or better than the small molecule corrector in all cases. But what we're most excited by was that when we combined the two, we got a super rescue of the current, of the surface density for many of these mutations. So far we've just been using NDUBs targeted to YFP as proof of principle, but this result made us get serious and really isolate nanobodies to the NBD1 domain of Cftr using a yeast library approach, yeast nanobody library approach that we obtained from Andy Cruz. we were able to isolate nanobodies and using FRET demonstrate with the full length channel. And so we armed this nanobody then with a dub to make it into an end-up, and asked whether we were able to rescue the whole cell current due to the mutations. This was work done by postdoc Zinara Shuja. So the wild type Cftr expresses robust outward chloride currents. And this is the most dominant mutate in F508del. The current is essentially gone. This is one for which the FDA has approved drugs. Here's one for which there are no drugs that are currently available. And that mutation also wipes out the current. The currently approved drug for treatment of CF is Cftr. It's a triple combination therapy consisting of 2 correctors and a potentiator. And this is what it does for F508. You were able to rescue some of that, and to a lesser extent for N1303K. But what happens is when you co-express, or when you express the end-up with that, you essentially rescue the current back to wild type. So the final experiment that I'll show here is that the gold standard for Cftr rescue would be these airway epithelial cells grown at the air-liquid interface, which grow this pseudostratified layer with ciliated cells at the epithelial layer. The wild type Cftr, you'll find it heavily staining the apical surface, and that is gone for cells obtained from a F508 patient. Lumacaftor, the FDA-approved drug, rescues it to a partial extent, but with the end-ups, we essentially get a full rescue of the trafficking to the apical surface. All right, so we've developed this approach, end-ups, that enables selective deubiquitination of target proteins. And we've shown it that it provides a robust general approach to treat diverse trafficking deficient ion channelopathies. And for what I don't have time to show here, the ubiquitin code is very complex, and we are interested in utilizing this approach to help us decode that in living cells. Okay, so I'd like to thank Scott Canner, Zunaira, and Ananya, who did most of this work, as well as my funding sources and collaborators. So thank you.
Great, thank you, Henry. Fantastic work. We have one question in the chat box. Can you phenocopy with proteasome inhibitors?
Okay, it's a great question. All right, so it's a great question, and it's the answer is a bit nuanced. All right, so with a proteasome inhibitor, you basically inhibit the degradation of all the proteins. You prevent those proteins from being degraded inside the proteasome. And what you're going to end up with are proteins that are highly ubiquitinated. For ion channels, that's actually problematic, because even if you have the protein there and it's highly ubiquitinated, it's typically inside the cell. It doesn't make it to the cell surface. And so this is actually a great distinguishing feature between these end-ups and the proteasomal inhibitors, in that, first of all, we do it with selectivity to a target of choice. And second, we strip the ubiquitin from them, and that is necessary for getting that channel to the cell surface.
Great, thank you. I might ask a question, if I may.
Please.
So, PROTACs, which are small molecules that are, so they're like heterobivalent, that will bridge your targeted molecule, like in this case, an ion channel, to endogenous ubiquitin ligases. Now, these are exciting from a therapeutic point of view, because they can get into cells,
Yep.
In your case, with the nanobodies, I don't know if you've got some clever ideas about how to deliver these. Maybe you can comment on that, or do you think that this approach is sort of a good first step towards prioritizing PROTAC development?
Great question. So thank you. So the short answer is we think we think that there is both a biologics play and a potential small molecule play for this technology. And for biologics, I mean, you're right, the delivery is the challenge, and you would have to think about a more gene therapy type of approach using a viral vector to deliver it. And the viral vectors are continually being developed, and so the idea is that hopefully you should be able to, at some point, be able to deliver and end up using a gene therapy in some way. And it's a really interesting question about the PROTAC analogy, because in a sense you can think about doing almost the anti-PROTACs, right? So it's a targeted protein stabilization approach. And there is actually a startup that's doing exactly that, trying to recruit deubiquinases to stabilize proteins.
Great. Thanks, Henry.
Thank you.
I'll remind everyone to ask your questions in the chat box. We'll move on to the next presentation from Markus Delling. Thank you, Henry, very much. So I know Markus well. He's residing at UCSF. He earned his master's in chemistry and biochemistry from the University of Heidelberg in Hamburg, Germany, before doing his Ph.D. in neurobiology at the Center for Molecular Neurobiology in Hamburg. He performed his postdoc training in David Clapman's lab at Harvard University, where he was also a Harvard instructor before joining UCSF, I think around 2016. Maybe that's like maybe half the time you've been enduring COVID in your lab, almost. So his lab studies the molecular mechanisms of how primary cilia utilize ion channels and GPCRs to sense their local environment and control important processes like left-right symmetry. And his long-term goal is to understand the signals that activate primary calcium channels into ciliary calcium channels, and to identify resulting downstream ciliary dependent signaling cascades. And they use a wide variety of approaches, including mouse genetics and RNA sequencing, cutting edge primary cilia calcium imaging, electrophysiology, biochemistry, really a range of different approaches to solve fundamental problems related to ion channel biology and human disease. It's great to have you here with us, Markus. Looking forward to your presentation. Thank you.
Hey, Mike, thank you so much for that nice introduction. And let me share my screen. Yeah, so let's go here. Yeah, thanks, Mike and Lily for giving me the opportunity to present our work here to this illustrious group of people. And yeah, so as Mike already pointed out, so my lab tries to understand the electric signaling of primary cilia, but I thought for this crowd to give a more ion channel centric presentation and really just describe in more detail the reductionist approach that we use to characterize channels which are really hard to get to because of their location. And I think this crowd here also does not need an introduction to trip channels, just as a broad brush. So my lab is interested in a subclass of trip channels that are called the polycystins, and obviously the founding member of the trip channels, the mammalian trip channels, is TRPV1 that was cloned by David Julius as the capsaicin receptor. And while
Markus, I'm sorry, this is Anna. We're not seeing your slides.
Oh, sorry. I'm very sorry. Thank you. Let me go.
Yeah. Yes, perfect.
Okay, sorry for that. Well, you guys didn't miss much. Let me go back here. So all that I was showing so far was kind of this, the overview of the trip channel superfamily. and TRPV1 as the founding member of the mammalian TRP channels that was cloned by David Julius as the capsaicin and temperature sensor. And the ion channels, or the group of channels that we are particularly interested in, are the polycystins. So as Henry pointed out, so a lot of channels, they traffic to the plasma membrane, and you can easily record them in wholesale recordings. However, there are several channels that localize to subcellular compartments, and in particular, TRP channels are notorious for localizing to subcellular compartments. And I want to share some data on the mucolipins that are endosomal channels, and how we use the approaches that other researchers have used to deorphanize the function of mucolipins, and how we can apply them to the group of polycystins. So what makes the polycystins in particularly difficult to study are actually two things. One of them is the localization to primary cilia, which are tiny hair-like structures that emanate from almost every cell. And the second is that they seem to form this unusual heteromeric complex where the TRP channel, so the PC2, forms a heteromeric complex with a PC1 subunit, which has 11 transmembrane domains and a long extracellular N-terminus. And so this N-terminus can get cleaved at an autoproteolytic cleavage site called the GPS site, but the crystal structure, the cryo-EM structure that was published 3 years ago kind of tells us that the last 6 transmembrane domains of PC1 form this intricate complex with 3 PC2 subunits shown here in blue, and somehow this doesn't move. So what makes PC1 and PC2 particularly interesting to study, except their weird heteromeric complex, is that mutations in PC1 and PC2 lead to autosomal dominant polycystic kidney disease, ADPKD, which is actually the most common monogenetic disease in humans. And it affects anywhere between 1 to 400 to 1 to 1,000 people. So there are roughly 12 million people worldwide that suffer from ADPKD, and the phenotype of ADPKD, so to say, is that over time you develop cysts within your kidney, but also other organs, these fluid-filled cysts that really impair your kidney function, so that you ultimately need a kidney transplant or kidney replacement. So 20% of all kidney transplants in the US are due to ADPKD. So how do these fluid-filled cysts actually form as a consequence of a mutation in an ion channel? Well, the short answer is we still don't know. So the idea is that these polycystins, they are expressed in this hair-like structure, the primary cilium, that somehow points into the lumen of a nephron. And these cilia presumably sense something. That signal that they sense is not really known. And somehow, when these channels that are when the polycystin channels in the cilium don't function properly, there is some weird signal occurring that results in proliferation, so that you get selective restricted proliferation along the nephron, and you develop these fluid-filled cysts. And here is an example of a kidney from an ADPKD patient. And just to give you the idea of the scale, here you see the hands of the surgeon, and here you see an individual fluid-filled cyst, so they can get pretty large. So obviously, this is just to set the stage, is so why we need to understand the function of this polycystin complex in primary cilia, ultimately to understand how ADPKD it develops, and ultimately also to develop novel therapeutic approaches for this, maybe using even approaches that Henry was pointing out. So the reason that we don't really, I would argue, so the reason that we don't really understand the molecular underpinnings of ADPKD is because we don't really had methods to reliably record that polycystin complex. So they localize to this primary cilium, which is extremely difficult to study. It's about the size of a single bacterium. And Paul Decane and Steve Cleaner and also my lab, several groups have now established methods to record directly from primary cilia. However, it would be just nice to also have a whole cell assay that would allow us to measure the impact, to measure the polycystin complex, just simply to do high throughput screens for small molecule activators on N inhibitors, and also just simply to understand the biophysical consequences of the mutations that cause ADPKD. And so what actually caused some confusion in the field is that simply when you overexpress PC2 in HEC cells, it doesn't really give you any current. And this is actually, as most of you know, pretty common for compartmentalized channels, that either there is a lack of membrane trafficking or a lack of activation simply when you overexpress them. So the goal that we took was just simply to do a very reductionist, approach, a systematic reductionist approach, where we wanted to kind of dissect these different possibilities of why simple overexpression of PC2 does not result in currents. So and obviously, what I haven't really mentioned so far is that PC2 is not the only member of the polycystin family. So there are two other members, so that are called PC2 like 1 and PC2 like 2. And they are actually 5 different members in the PC1 family that are called PC1 like 1, 1 like 2, 1 like 3, and PKD REJ. So they all share the same membrane topology, and the main difference between these different isoforms is that they differ in their extracellular N-terminus. And so the genetic and functional evidence is that these different PC1 and PC2 members can form different complexes, which makes it even more difficult to understand when you have to think not only about an individual homomeric channel, but also different combinations of heteromeric complexes under the physiological settings that you are studying. And so for PC2 like 1, we have evidence that it interacts with PC1 like 3 and 1 like 1. And at the end of the presentation, I will show you some evidence that we also established our assay to study now PC2 like 2, which is a channel that has not been published, or the biophysical properties of that channel have not been published. And I'll provide evidence that it forms a functional channel with PKD REJ. And exactly for this reason that they are so difficult to study, they are also on the they are qualified as IDG targets, as dark matter ion channels. So, and so this is just, so I was mentioning earlier that the closest homologue to the polycystins are the mucolipins that are in endosomes, and also the function of the and the regulation of the mucolipins was not really known for a long time. Just as you can see, appreciate here, when you overexpress them, they localize to endosomes, and direct electrical recordings of endosomes were not possible for a long time. Only Hao-Ching Xiu's group using pharmacology to enlarge these endosomes allowed them to record from the endosomal membrane. So the approach that they took to kind of deorphanize the mucolipins was just simply to retarget them to the plasma membrane so that you could use whole cell recordings. And also initially the trick was just to use a gain of function mutation that would overcome the lack of activation or the lack of potential second messenger or the lack of second messengers that may be specific to endosomes and are absent in the plasma membrane. So as you can see here in those papers, so when you target, when you introduce the gain of function mutation in TRPML3 and you target it to the membrane, you can actually nicely see an inward current. So clearly that was the trick to understand the regulation. And ultimately later on, it also allowed the screen for small molecule activators of this complex that are now in clinical trials, I believe. So the work that I'm presenting now is from two very talented researchers in my lab. Kodachi Ha is a postdoc, and when she was in my lab, she was a technician, but she was super motivated technician, and she's now a grad student here at UCSF. So, so the approach that we took is just simply, well, how can we systematically dissect trafficking versus activation defects or problems as a result why we cannot really record this complex in the plasma membrane? Well, so the approach that we took here was maybe we can force this complex to traffic to the plasma membrane. And we chose two approaches. We either replace the endogenous signal peptide with the strong secretion sequence, the kappa IgG sequence, and we also introduced an extracellular HA tag, just similar to the approach that Henry was telling us about, so that we can quantify surface trafficking of the complex. And the other approach that we took is just simply that we replace this huge extracellular N-terminus with the N-terminus of the P2x12 GP receptor, which is a GPCR, and so this P2x12 and a short N-terminus of P2x12 has been shown to also promote surface trafficking. And at the same time, we also introduced an extracellular FLAG epitope in PC2 so that we can really monitor surface trafficking of both proteins just simply by using a surface HA and surface FLAG staining. And as you can see in this image here, when we do surface HA and surface FLAG staining using our this kappa PC1, we actually see we see strong surface membrane fluorescence when we co-express PC2 with kappa PC1. However, the wild-type PC1 shows only very minor, if at all, surface expression, indicating that really the kappa IgG secretion sequence is the trick. And to a similar extent, the construct that we call PC1 delta NT also nicely traffics to the plasma membrane, and it drags PC2 along. And sorry for this. Hello?
Yes,
Yes.
we're here. We can hear you.
Yeah, somehow I can't. My computer just froze. Let me see. Oh yeah, no. Here we go. So yes. And so here we quantified surface HA and surface FLAG staining, and you can nicely see that the two proteins co-traffic together when co-expressed, but only our modified PC1 constructs were doing the trick. And here is the surface staining. So this is HEC cells. You see nice membrane staining. And when you transfect IMCD cells that form a cilium, you still see that the complex localizes to the primary cilium, as highlighted here with acetylated tubulin, indicating that we don't really change the endogenous properties of the channel when the cell is instructed to form a cilium. So, and so this goes also back to this gain of function mutation, the proline mutation that we found in the mucolipins to result in a constitutively active channel. So Yong Yu's group had published that when you introduce a point mutation at the similar or at the corresponding mutation in PC2, you also get a constitutively active channel when you use oocytes. However, we also had obviously tried the F604P mutant in hex cells, and we never saw any similar currents, which suggested to us that, yeah, I mean, clearly there is also a surface trafficking problem. So what happens now if we overexpress? What happens when we now overexpress? Can we record from cells where we see the complex in the plasma membrane? And so the answer is, when we overexpress our membrane-targeted PC1, PC2 complex, for wild-type PC2, the channel still remains silent in the membrane. However, when we co-express the kappa, the membrane-targeted channel, and introduce the gain-of-function mutant, we see a very nice outwardly rectifying current. And we really need the PC1 subunit as overexpression of PC2, F604P, and hex cells does not result in any currents above background. And as I pointed out, it looks like a trip channel. It's an outwardly rectifying channel. So so we next asked, well, I mean, how could this channel be potentially regulated? And remember, I told you that we made two constructs, the full-length PC1 and the one where we replaced the N-terminus with the P2x12 N-terminus. And to our surprise, we realized that only full-length PC1 results in a constitutively active channel, while the lack of PC1 N-terminus, the channel remains non-functional, despite comparable surface trafficking of the two. So I think here really, I hope you can appreciate now the reductionist approach that we took, that we can clearly now differentiate between a trafficking problem versus an activation problem. So this encouraged us, so this made us hypothesize that this N-terminus, which is decorated with all these adhesion domains, somehow is critical in the sensing of the channel and presumably for sensing of primary cilia. So in order to narrow down the region of this N-terminus that might be critical for the PC activation, we looked at the other PC1 members and to determine whether they would also traffic to the plasma membrane using our kappa IgG approach. And as you can hopefully appreciate in this slide here, when we co-express PC1 like 3 together with PC2, we also get very nice surface staining for PC1 like 3. And here in this cell, there's so much surface trafficking that you will stain both the plasma membrane and the primary cilium. And so we went ahead and made different chimeras between PC1 and PC1 like 3 to determine the effect on channel activity. And I hope, as you can appreciate here, so when we co-express each of these PC1 members or the chimeras together with PC2 F604P, we only see constitutively active channel for the PC1. However, the PC1 like 3, despite trafficking to the membrane, still does not result in a constitutively active channel. However, when we swap the N-terminus of PC1 onto PC1 like 3, we can restore channel activation. So this made us hypothesize that this N-terminus of PC1 is really critical in the activation of the complex. And I don't have time to go into that, but we narrowed down the chimeras and we found that it's a C-type lectin domain that is the minimal domain that's required for this activation. So as I had pointed out, so the strange thing or the weird thing about this polycystin complex is that the N-terminus gets proteolytic, it undergoes autoproteolysis and potentially could be released as a ligand. So we tested whether when we express this protein in HEK cells and purify it. So this was done in collaboration with Erhu Chow at the University of Utah, and apply N-terminal fragments to our cell lines that express PC1 delta N-terminus, whether those ones would also restore channel activity. And indeed, we can show that when we perfuse cells with the N-terminal fragment, we can also restore the complex. And when we boil, when we denature the fragment, the activation is diminished. Okay, so I think we are just getting slightly to the end, and I just wanted to share two unpublished results with you that we were now able to obtain using our novel assays. And so one of the strange things that came along while we were doing the study is that when we record the polycystins from the ciliary membrane, we see functional channels in the ciliary membrane. So these are wild-type polycystin channels. However, as I was showing you earlier, when we overexpress wild-type polycystins in the plasma membrane, these channels remain silent and are only active when we introduce the gain-of-function mutation. So the question really is, why is the wild-type polycystin complex active in the cilium but inactive in the plasma membrane? So this made us hypothesize that there might be cilium-specific second messengers that control activation of the channel that license the polycystin complex to be active only in the ciliary membrane and not in the plasma membrane. So long story short, we, in collaboration with Jeremy Reiter's group that had done lipidomics on primary cilia, we tested a bunch of lipids that are specifically enriched in primary cilia, including some of the PIPs and oxysterols. And as it turned out, we can now also activate the wild-type polycystin complex in the plasma membrane when we apply a specific oxysterol, which it's a dihydroxy cholesterol 7-beta-27. When we apply that in the patch pipette, so from the cytoplasmic side, we can also see, record the polycystin complex that has similar properties as the F604P mutant. And this effect is very specific to one particular oxysterol, while it's not activating, while other oxysterols are not capable of activating the polycystin complex. So just to summarize, this work. So we think the polycystin complex has a dual regulation. The N-terminus somehow is the sensing domain that's critical for activation of the complex. And then there are these oxysterols that ensure activation of this complex only in the primary cilium. And so the last slide that I want to share with you is this dark matter ion channel PC2 like 2. And so we tested a bunch of the different PC1 isoforms and co-expressed them with PC2, where we also have introduced this gain of function mutant. And it turns out that PC2 like 2 forms a functional channel only when you co-express it with the PKD REJ domain. So we are now trying to understand in which cell types this complex may function, and so far we haven't really confirmed a ciliary function of this complex. Okay, so I want to just finish by thanking the people that have done the work and our collaborators and my funding sources, and I want to thank you for your attention.
Great. Thanks for the great talk, Markus. I'll remind everyone to add questions in the chat box. I'll start. I had a couple of questions. The lectin binding, you hinted at the lectin binding domain, because the PKD family having this unique N-terminal domain is kind of interesting, right? So you suggested that the lectin domain is an important component of that. Do you know what sugars bind to it? I mean, it implies almost like there's some carbohydrate entity that's important for the function of the channel. Have you thought about maybe putting glycosidases in your patch pipette and recording,
that's a very, so with Erhu and Chow, we are now screening for carbohydrates that may selectively bind to that C-type lectin domain. And yeah, you're absolutely right. So the interesting thing about, and I didn't have time to go into that, is this PC2 channel and the mucolipins too. They have this top domain, which is this unique extracellular domain that sits above the channel on the extracellular side. That's also heavily glycosylated according to the cryo-EM structures. So we think that C-type lectin domain may actually bind to the carbohydrates that are expressed on the top domain of the polycystin complex.
I see. I know some biochemistry has been done on cilia. Do you think that there are maybe some factors present in some of those mass spec data from cilia that may bind the complex carbohydrate? Like, do you see the complex carbohydrate as being sort of a sink for molecules to bind to, or perhaps like a regulatory thing?
That's excellent question, Michael. Yeah, I mean, we haven't really. Yeah, I think that would obviously be very cool to do more proteomics on also using the N-terminus as a bait, and we haven't done that. But I agree with you.
Gotcha.
Can I ask a question?
Please. Henry.
Okay. So yeah, great talk, Markus. I was wondering what's known about how the channel is specifically targeted to the cilia first, and second, whether those mutations, whether some of those mutations are actually trafficking mutations or not.
Exactly. No, that is exactly. So yeah. So first part of your question. So the N-terminus in PKD1. So Steve Sommel's group had shown that there's a kind of a ciliary targeting motive in the N-terminus of PC2. How and whether PC1 by itself traffics to the cilium is still a matter of debate. And I mean, we are doing these experiments more systematically now. And the second question is, yeah, I mean, you're absolutely right. So 20% of all ADPKD mutations are missense mutations. And clearly those missense mutations will either affect trafficking or channel activation. And I mean, Paul just had a paper, I think in PNAS last year, where he showed that a mutation, an ADPKD mutation in PC2 affects activation and not trafficking. And we are also now classifying mutations, ADPKD causing mutations in PC1, and we can see that some mutations affect trafficking and others affect channel activation. So clearly we will you know, there are like 2,100 potential ADPKD mutations in PC1. So obviously it's a daunting task to test all of them. But so we selected, you know, in different regions of PC1, and we can show that, yeah, clearly they fall in two groups.
Great. Thank you.
Great. Oh, by the way, Henry, we generated a bunch of constructs, you know, that I think we might have already sent you for generating nanobodies, and some of those contain like PKD1L2s, like flag and a variety of epitopes. Might be worthwhile to really generate some nanobodies against the PKDs, you know, in that way.
Absolutely, yeah.
So it'd be great. It'd be great for you guys to talk. I want to say a huge thanks for you guys for joining and giving and sharing your science. Really fantastic talks. And I'll